So good afternoon, everyone. I believe some of us are coming from Singapore and Taiwan, Malaysia. And all right, so I am Kai Ling, the Regional Field Application Scientist of Azure Biosystems. This is the final session of session 3 of this Western Blot e-learning workshop, co-hosted by Apical Scientific and Excel Scientific. This session we will be talking about the new normal in quantitative Western blot, which is the total protein normalization. First, for those who just joined today, let me introduce Azure Biosystems to you. We are a company based in United States, California, very near the Silicon Valley. We are a team of highly experienced scientists, engineers, and entrepreneurs dedicated to accelerating your science. Our mission is to give you the raw data that you can rely on using the simple workflows that you can trust. Azure Biosystems is based on innovation, is co-founded by Arnold Shifji, our CEO, a venture investor and entrepreneur, and also Dr. Diping Che. Dr. Diping is our executive VP of R&D, so you can look through his past experience. So he has a lot of experience in Illumina and Alpha Innotech, a brand that we're quite familiar of. This is our timeline. Our company founded in 2013. The core members of Azure Biosystems are all coming from Alpha Innotech. So these are the few key instruments that we have right now on the market. The first one will be the Sapphire Biomolecular Imager, which is the industry first hybrid CCD imaging system and also / laser scanning system. And also our Azure imaging system, mainly for Western blotting purposes and gel documentation, and also qPCR system as well. Right, so let us recap what have we talked about in the previous 2 sessions. Firstly, we talk about the principles of each of chemiluminescence Western blotting steps, from the gel electrophoresis, transfer, blocking, up until the antibody detection, with 2 main category or 2 main methods that is used right now, which are chemiluminescence and also fluorescence method. We also talk about the signal linearity between these 2 methods, the quantitation power, and also which one is best fit for your experiment needs, because each of us have different application and different kind of question we want to answer. And next, how would you want to image your blot? Then we talk about the chemiluminescence. We'll be using the CCD chips. The CCD chips are different in terms of pixelation and also dynamic range, and also the capacity that it can behold. And for fluorescent method, we have talked about the excitation light sources and between the differences between RGB and near infrared signals as well. And last week, in session 2, we walked through the chemiluminescence Western blotting protocol in the detailed format, and also look at some of the general troubleshooting tips. A quick look at the fluorescence Western blotting protocol as well. And finally, we have proposed a workaround for you to improve your chemiluminescence quantitation power with total protein normalization. Today, we will go in depth into total protein normalization. Okay, so this is our agenda for this session. Firstly, we will talking about total protein, housekeeping protein, and total protein normalization. Then next, if you want to go for total protein normalization, what are the criteria you need to choose? You need to have in choosing suitable total protein stains that fit into your own protocol. Then we walk through a total protein stain protocol with Azure Red, which is a total protein stain we have in Azure Biosystems. Then we look at how you're going to analyze the data to get your total protein normalization analysis. And finally, how you're going to publish this total protein data as a replacement to your housekeeping protein data. And also look at some application note regarding this as well. Okay, so first, let's look into quantitative analysis in Western blot. So for so many years, researchers has been using housekeeping proteins such as like actin, tubulin, or GAPDH to normalize the Western blot loading. So these are usually detected alongside. For example, you have protein A and B. So let's say your protein B is your housekeeping protein. Your protein of interest, let's say protein A, will be detected alongside with your target of interest, either in parallel, such as in this Ferguson Western blot, or you do stripping and reprobe process to detect one protein at a time. So you can determine the relative target abundance in different samples across different all these lanes. So this is achieved by dividing the signal intensity of the target, let's say here, by the housekeeping protein in each lane. So in terms of data analysis, we calculate the ratio of, let's say, A divided by B to account for variabilities in both sample loading and transfer efficiency. So this is how it looks like in terms of housekeeping protein. You have one housekeeping protein, maybe one or more, depends on your experiment, right? So this is one. In total protein stain, stories are different. This is no longer a single band, like you can see over here on the left, housekeeping proteins bands. These are multiple bands. That's why it's called total protein stain. But we're going to use all these bands, okay, in this lane to become the normalization factor, okay, for your protein of interest. So the differences is quite obvious here, one versus many, okay? Right. As an important component of accurate quantitative Western blotting is the normalization of the band intensity obtained from the target protein, okay, against an internal reference that doesn't change with the experimental condition, but most of the time, it is not that case. So this so-called the control, it will change. It doesn't change with experimental condition, but it will change proportionally to the protein amount that we load into the gel. That is an ideal situation. Okay. So traditionally, all these housekeeping proteins are the proteins thought to be consistently expressed across different sample types. So they have used such a reference to control sample loading across entire membrane. Okay. But since there are multiple steps lie between protein determination, what I say, probably Bradford assay before load it into the gel, then to the final step of the Western blotting, which is a visualization of the blotted proteins. There are four factors that can prevent equal loading on the blot. Firstly, I didn't put it on the slide here, but I just mentioned it verbally. Firstly, error in the protein determination due to incompatible lysis buffer compound. Second, inconsistent protein loading into the lane. For example, you have bubbles in the pipette tips or some pipetting errors. Third, inconsistent protein separation in the gel. So when you run the electrophoresis, okay, so the protein separation between the first lane and the second lanes are different. It might be due to the inconsistent protein entry into the gel matrix. If you have the stacking gel, when the stacking gel entering the resolving gel, it might be different. So you might see some bands that are not at the same molecular weight as you expect them to see. Then a final factor will be the unequal protein transfer efficiency across the membrane. So this will be about the transfer part. Okay, so because there's so many steps in between the Western blotting protocol, so as a single indicator will be quite risky in this sense. All right, so when we rely the housekeeping protein to signal us, to tell us that, okay, you have a proper sample loading, your transfer is good, your gel running is very good. So these are very I'll say not so kind of like a lot of things you need to be aware of, like say highly variables. And then another very important thing is the so-called housekeeping proteins are not really housekeeping in that sense, because we do find out that in certain publication or maybe in your own experiment, you realize that these housekeeping proteins do change according to the experimental conditions, even sample type, cell type, disease, treatment type, even the time point that you harvest, even the day that you harvest, or even the weather, ambient temperature, so on and so forth. So there's a lot of factors actually affecting this. So, and as we expect them to be all highly expressed, since they're highly expressed, so we assume that they are similar amount. This also contribute to somewhat we might be seeing in this future is a narrow linear dynamic range. Means you usually will saturate the membrane or saturate the CCD imaging system if you're in your usual protein loads. So you have to be like consider optimize it, like how much you need to load into the gel, so it wouldn't saturate out the protein or saturate the blot, so render the image unusable. So there's a lot of factors that here you need to consider. Okay. But so here we list on the left, we'll be listing the drawbacks for using housekeeping protein for normalization. So firstly, inconsistent expression across samples and condition. It happens from time to time. It doesn't mean that it happens to all of the people. It depends on your sample as well. Okay. And it could be easily affected by experimental treatments and condition. And then due to these highly abundant or high expression, they have narrow linear range compared to your protein of interest. So this is something that very common. Okay, so thus you're having the risk of saturating your blot later during the imaging step. And as we talked about in the previous two sessions, we know that chemiluminescence is really good in detecting a single protein on a blot. It's very sensitive. It's very good for single protein. But if you try to detect like more than one protein at the same time on a blot, you will face certain difficulties. For example, you want to detect a phosphorylated protein and a total protein that couldn't be separated by gel electrophoresis based on their molecular weight, because both of them are having similar molecular weight. Then you have to go for either chemiluminescence method, strip and reprobe, or you do it in parallel gel. Okay, so these two have their own, I'll say, perks and also advantages and also major drawbacks as well. So for those who do post-translational modification studies, I will highly recommend you to go for fluorescence method if you can, if you have such equipment or capability or do so. This will save you a lot of time and kind of control a lot of situation as well. Right. Lastly, housekeeping protein also will interfere with your protein of interest imaging, because we often choosing housekeeping proteins that is having different molecular weight than your protein of interest. Like for example, if I want to study actin, but actin, there are different types of actin. I cannot choose the actin that is very near to the protein of interest as a housekeeping protein. So I need to choose something else to become the housekeeping protein. Right. So there's a lot of matching combination to be considered before you carry out your experiment. So, and finally, it contributes to, I would say, the difficulty in visual identification if you want to detect like one at the same time. So there's a lot of things to consider, even antibody choices and blah, blah, blah like that. But I will not go into details. Okay, so we know about housekeeping protein drawbacks for normalization, but this will only happen if you do have this kind of situation in your experiment. If your housekeeping protein has been working well, it has been consistent throughout your experiment, and even sample types and conditions, then that is pretty good. That is super good. Okay, then stick to it. All right. Okay, but if you realize that, okay, you might see some abnormalities, or not say some changes, or that something you cannot be explained, then you might need to consider to opt for total protein stain as your normalization control. So based on these drawbacks, there are several alternatives for housekeeping protein as a normalization control. Firstly, of course, you pick a different housekeeping protein. Okay, for example, like the DJ1 protein. All right, so all this information available in this publication, Moritz, published in 2017, where the author summarized, kind of compare different total protein stain and also comparing what, saying what is the drawbacks and what the advantages of housekeeping protein and also total protein stain as well. So I will highly recommend you to go through this publication, kind of see what is happening right now, and then what are the good and bads of different staining and different techniques and so on and so forth. Okay. All right, so let us go back to the alternatives. DJ1, all right, DJ1 protein. It is considered more stable protein, but it also has a varying expression issue, and then the immunosignal saturates around 20 microgram of protein loads. Okay, so there's no really a perfect housekeeping in this, and you have to choose it according to experiment, right? So what if I just want to choose more than one housekeeping protein as a loading protein, so you can lower the probability? Yes, you can do that. So, okay, you can average out the more than one housekeeping protein as a loading control, but of course, it will your cost will be increased. The time also increase as well, because bear in mind you need to, the combination of primary antibody and secondary antibody also increase, because you see there's only that many hosts and species that we have to generate antibodies. So there's a lot of pairings that you need to do and need to consider if you want to add on one more housekeeping protein in your experiment. Okay, then if let's say, okay, these two are not suitable, then how about I just want to characterize my current housekeeping protein before the experiment? Yes, you can do that. One way to do that is pull your samples, regardless from what kind of group, pull them together, get a so-called pooled samples, okay, run the samples in a serial dilution, okay, create a standard curve, okay, and then see that whether the expression changes or not. Or the easiest way is you just test it out. Just run it through, just randomize them, each of these samples, okay, test it out together, see whether it's nice or not. Okay, whether it's showing consistent housekeeping expression. So there's a lot of ways to do that. You can search online on how to do this. So I will not go really into details on this, because this will talk about experiment design and blah, blah, blah like that. Okay, right. So if you're capable of doing thorough characterization, okay, but of course, do remember that you will increase your cost and time and all the preparation for you to quantify it properly. Okay, right. So there's also another method that we adopt from qPCR, which is spike in equal amounts of standard protein into each of these sample, so we can use it as a control, normalization control. You can do that as well. Okay, but it will also having issue, because you need to choose what kind of standard protein you want to put in. You have to characterize it properly as well. So there are additional steps to do that, right? So since, let's say I'm just choosing another protein for alternatives, for housekeeping protein alternatives, not really, maybe some of them are not really feasible for some of us. So what then they propose, the community proposed different kind of alternatives. So firstly is, what if I just want to visualize the total protein on the gel before I transfer? Well, visualizing total protein itself is a really good idea. So, so which step we want to visualize? If you want to visualize the total protein on a gel, then you're omitting the transfer efficiency factor out of this normalization control. So I wouldn't say this is the best method unless your Western blotting is not really required at such accuracy. Then I'll say this is okay. Okay, right. So if you want to go for really quantitative Western blot, then the next alternative they furthermore propose is, I just want to visualize the total protein on a parallel gel. So it's a similar thing like the previous suggestion, like visualizing on the gel before transfer. If you visualize total protein on a parallel gel, you kind of omitting, it's not really representing your plot anymore. That's the issue. It's the same issue as you like to visualize a protein B or housekeeping at a parallel gel. It's the same thing. So it doesn't it doesn't improve it, to be honest, right? But it's just that you have a total protein, which is better. But we want the ultimate accuracy. Therefore, they further propose this: visualize the transferred total protein on the blot. Therefore, if you look at all the total protein stains on the market, most of them, they will put on this step. they will encourage the stain itself will be allows you to visualize the transfer protein on the blot. Okay, and then because this is currently the best technique, the optimal, the easier one you can go, and the most convenient way to solve the drawbacks that we observe over here. Okay, I hope this is clear for you. Right. Yeah, here. So this is the most convenient one. Okay, so now let us go to total protein normalization. Like I said, with this method, instead of trying to find a protein that can represent the total amount of samples that transfer on the membrane, the total protein is measured on the membrane directly, and this value will be used as a denominator when normalizing your protein of interest. Total protein normalization has a wider dynamic range, okay, larger linear range, dynamic range, because it allows the detection of lowly abundant protein, you can see here, with the very faint bands, and also highly expressed housekeeping gene, including housekeeping gene as well, right? But you having lesser risk of oversaturation. That is one of the perks. It has smaller technical variation because you avoid lane-to-lane variation that inherent in Western blotting. Because you see everything, you see all the bands in a single lane. When you compare to the other lane, you will notice the differences. The protein bands will not more slightly similar, but the intensity. So let's say if you have a pipetting error, you will see it very clearly in total protein normalization. Okay. Right. So next thing is, yes, a lower variability. Okay. He has a smaller technical biological variation. The biological variation here is since because the whole entire protein population of the sample is less susceptible to change than a single housekeeping protein. Like I said, housekeeping protein is a single protein band that we use it for as a normalization control. But total protein, we're using many bands. So of course, the variability, the variation will get reduced, the error, chances for error also reduce. So that is the root of why total protein normalization is better in a general sense. Okay, and it has minimal changes according to experiment condition because we are staining all of these protein. We are not relying on one single indicator. And then it also takes shorter amount of time to take this image because especially in Chemi Western, no, you don't need to stream and report the blot anymore. Okay, and total protein stain also can be used to monitor protein transfer. Okay, so you can do additional QC on your blot. You can use it to verify your transfer is complete and free of artifact as well. So you can see the bands are nice. Okay, so there's no bubbles in between. Right. So there's a lot of total protein stains available on the market. You can use it to stain the gel, or you can use it to stain the membrane. Right. And then it provides in summary, it provides a lower variability and cleaner data than housekeeping proteins. But there's one also important thing is, just like if you're choosing different housekeeping protein, you need to ensure this staining you use is going to be compatible with your antibody binding and also detection method. So it will not interfere with your protein of interest detection. Which is our ultimate aim of doing Western blotting, right? Okay. So in that, anyone performing Western blotting can benefit from total protein normalization, and it has become the new standard required by journals and for Western blot quantitation. They kind of promote it because it is definitely better than housekeeping protein as a normalization control. And so this is the best time to switch or even to transit from using housekeeping protein for normalization. Okay, so if you recognize the benefits of doing total protein stain, so you're just interested in it, you might be wondering, so where should I start? So the first thing is you need to know how to choose a total protein stain. Here's a list for you to consider when you're choosing a suitable total protein stain. Firstly, of course, toxicity. Okay, shouldn't be toxic to human. And secondly, does it work with your sample type? Is your sample type cell lysate or biological samples? So there's something that you need to look into the total protein stains and see what kind of sample they are best working with. Of course, staining, any kind of staining that gives you signal, you need to look for the signal linearity. Like we talked about in the previous sessions, signal linearity is very important because it should reflect what you have loaded into the lane or the blot. Because by the end of the day, we want to do quantitative analysis. And with this, normally total protein stains will give you a guideline, or they have tell you what is the optimal range of protein lysate concentration that it can detect within that range. Okay, so this is easier to work with. And of course, sensitivity. What is the detection limit per spot with a lower background and no speckling? You want something with clean background, okay, but you can able to see the protein bands. That is what you want to see. Then versatility and also compatibility of this stain with your current protocol. For example, does it compatible with your PVDM membrane, microcellulose membrane, or even the gel that you're using? Is it compatible to your downstream fluorescence or chemiluminescent Western blot immunodetection? Would it interfere with the signal detection? And do you have that kind of imaging system, for example, CCD camera, or even the fluorescent channel to detect it, or even the excitation light source that can activate it? So there's a lot of consideration here. And finally, of course, the very important one, does this stain giving out residual signal in the detection channel of your protein of interest? By the end of the day, you do not want this to interfere with your protein of interest imaging, because otherwise it will defeat the whole purpose of getting total protein normalization, right? And also, if you have special application, you might need to look at whether they have the reversible or destaining option, or is it a permanent staining on the blot? And finally, convenient workflow. We want it to be convenient to incorporate in your existing Western blotting protocol. It's because you want to replace your housekeeping or you want to start up. Of course, it must be something that's convenient, general, and easy to use. So these are the list of consideration. It's kind of like common sense. These you can look into it and then look for the protein stains that works with your experiment. Okay, so I will go into each of these criteria. Signal linearity, I'll be using total stain Q as an example. So total stain Q is from Azure Biosystems, where it says it is the ideal cell type for this staining is cell lysate. And then it's best working for high protein loads, for example, one ranging from 1 to 50 microgram per lane, okay? And the detection limit is 20 nanogram. All right, so if your protein load has been this concentration and you're using cell lysate, then total stain Q might be the choice of your total protein stains. Okay, so this is like I said, you consider about the criteria. Right, signal linearity, how you're going to look at it is easy. You having known proteins, for example, we have cell lysate. Okay, dilute it, load it in a serial dilution. Okay, then you plot the signal intensity. For this total stain Q is image is Cy3 channel, and plot it and make a standard curve and see whether you get a signal linearity or not. And then within the said detection limit, okay? Yeah, so if it's good, that means, yeah, it's safe for you to go. Okay, you can even request for trial kit, and then try on your sample to see whether it works for your sample. Right, so this is one of the criteria that you need to look for. Second is dynamic range. All right, so this is the paper I mentioned that published by Maurice, the tubulin or not tubulin, heading towards total protein staining as a loading control in Western blots. All right, so this is a figure we put out from this publication, mentioning the dynamic range of this staining that found on the market right now or available right now. So we slot in the two total protein stains that we have in Azure Biosystems. Like I said, total stain Q is working for cell lysate, and Azure Red will be working for biological fluid with different kind of dynamic range. Can you see? Right. So we have on this figure, you can see there are multiple stainings available right now. You can do Ponsil S, Cypro Ruby, Comexi, Amidor Black, other stain-free technique, or even using side dyes and epicoconone. Okay, so Azure is a type of epicoconone. Okay, so it's working like this. Then, so this is for you to consider what kind of staining you want to use that it fit into your protein load into each of the lane. Okay. Right. So versatility, like I said, what kind of membrane they're using, either you want to detect on the gel, what kind of immunodetection, do you have the imaging system, or does it having residual signal as well? Right. So I'll be using total stain Q as an example here. This is a fluorescent blot. Okay. We have the duplex, I'll say duplex detection here, and also the total protein stains in the green channel. So we image it at site 3, the green channel, and then this you can see that it's compatible with the near infrared detection of 2 channel without destaining. Okay, so the important thing is do not see any residual of the signals in these channels in the image, so it won't interfere your data analysis later on. And the reason of this is you need to look into the dye itself, the staining itself, what is the excitation emission wavelength. Okay, normally total protein stains that will utilize fluorescence because I say in the previous session, fluorescence will give you very linear signal. It is much more stable because unlike chemiluminescence is enzymatic reaction. Okay, fluorescence is very stable. Whatever you is only influenced by energy input, which is from your excitation light source, and also the dye itself. The dye means how much your protein is tagged or labeled. So it's very simple. That's the reason that a lot of total protein stain they use fluorescence. Okay. All right, so this is pretty much for fluorescence. Western blot is compatible. Then how about chemiluminescence? Yes, there are dyes like, for example, this here will be using total stain Q is also compatible with chemiluminescence Westerns. Here you see that they will not have any, it will not affect any ECL signal, as you can see here. So you can safely analyze your data, your protein of interest, and also total protein stain, and then quantitate it accordingly. Right. This is just one of the example, or few example for you to test out or even check the versatility of it. I will really advise you to acquire trial kit and test it out on your sample to see whether it works or not. Okay, if you have the imaging system. Right. So here additional categories like labeling step, which step they're labeling, what is the excitation channel that it needs, what are the binding types, and also optimal protein loads. So we have Azure Red and total stain Qs over here. Okay, we have also other dyes available on the market like stain-free, side dyes, and also revert. Okay, you can see that they are labeled at different steps. Okay, so the red color indicate that they might have high risk of errors here. So like I said, if we do preloading labeling, you kind of like, it's kind of like having not, you also, the loading error, transfer efficiency, and different type of factors is not counted into this control. So I wouldn't really recommend this, but it also has different kind of perks as well. It depends on your application. Right. So we have post-transfer labeling, pre-transfer labeling, which is in the gel, and so on and so forth. Excitation channel, like I say, mostly will be in fluorescent channel. Okay, like you say, like you see here, the green channels. Okay, 700 channels and so on. And some of them may need UV to excite it. So you need to see your digital imager whether it's compatible or not. Okay, so binding types, there are major 2 types of binding. So firstly is no chemical binding, which is based on binding affinity. It is reversible because it's not chemical bonded. Okay, so there are other permanent bond, which is like covalent bond. Okay, these are irreversible. So this is very good if you do not need to, wash off the total protein stain to do subsequent experiment, for example. Okay, and the optimal protein load, you have to check them out to see which one best fit for your current Western blot protocol, how much that you load into your gel. So yeah, This is one of the things. Okay, so you can see here, I want to highlight here. So some of them I label in red because it might be not favorable for certain people, but it doesn't mean that this staining method is bad. No. If you, let's say, you just want to purify protein and check the protein concentration or check the protein purity, for example, then you do not need all these things. Like I mentioned here in the Azure and Total Stain Q. Stain free is working fine because you just want to get it get a normalization control. This is working. These criteria will be working if you want to do accurate quantitative Westerns. All right. Western blot is a general technique. It doesn't mean that you have to do. It's only quantitative. There are other methods that using Western blot to analyze a protein expression. Okay. Right. So please look through this kind of criteria, different kind of consideration when you're choosing your total protein stains. Okay, so after you choose your total protein stain, you're going to ask, how do I incorporate this stain into my routine Western blot? Okay, so I'll be using Azure Red fluorescence protein stain to explain to you how it works. Okay, as an example. So to bear in mind that different stain will have different staining protocol and different staining requirement in time as well. So you need to check them out, look at their manual to see whether it fits into your Western blot protocol or not. Right, let's look at the list of material that you need to use in Azure Red. And of course, this is Azure Red fluorescent protein stain. And then you need high purity water, either molecule or distilled water, need ethanol to activate it, staining tray, shaking or rocking platform, and also digital imaging system with UV, blue or green channel. The reason for this is Azure is a type of epicoconome. The optimal excitation is here. You can be either at the UV range and also the green range, and the emission will be at the green channel. Yeah, so it's based on different staining or different stain dye that they use, the chemical properties of it. So the requirement for different what kind of channels and excitation will also varies as well. All right. Okay. So epicoconum is a very small, natural occurring fluorescence, a compound that reversibly binding to lysine, arginine, and histone residual in the protein and peptides. So it will yield an intensely red fluorescence product. And this unique mechanism will provide sensitive quantification of protein in 1D and 2D gel of all chemistry, and both tested on both PVDF and nitrocellulose blot. Okay, and it also provides unparalleled compatibility with mass spec as well. Yeah, let's look at the overview of the staining protocol. As you can see, this is a post-transfer staining. Post-transfer staining means you run the gel, transfer, and after you get a blot, okay, then we go to total protein stain protocol. Rinse the membrane, okay, staining, fixing, then washing, then you proceed with your imaging with the rest of the workflow. The details are listed over here. Okay, firstly, of course, you need to prepare the solution. So I would say there's some tips over here, like do just mix them properly so you do not have any precipitation or dust, or else it will result in speckling. Okay, and the washing one will be like this. You have your transferred blot ready, wash it into purified water for 5 minutes. Okay, so it will advise you to run the buffer off the base on the gel. If you, let's say, using the bromophenol blue, for example, run it off the gel, then before the transfer. Then when you transfer, there will not be any bromophenol blue that interfere with the transfer, and also subsequent staining. Okay, and one keynote here is do not allow the membrane to dry during the staining, just like our usual Western blot. Then we stain the PVDF membrane on the stain solution with gentle rocking, then fixing the solution with acidification with gentle rocking. So the blot will now appear as a green color blot. Then you need to wash off the blot, the excessive dyes with 100% ethanol until the green background is completely removed. Then you can either dry it if you want to image it straight away, or you carry on with the rest of your Western blotting protocol, which is blocking, primary antibody incubation, secondary incubation, washing machine, and staining. Yeah, so you can do that. So this is compatible with two different types of protocol. Then the imaging part will be detect the fluorescence at the green channel using standard fluorescence scanner or CCD camera system. And the compatible excitation light source will include green channel and also UV light. Okay, right. So troubleshooting here, we will, like I say, total protein state, because it's staining all the proteins, total proteins in a single lane, or not a single lane, on the lanes. So if you have any artifacts, it will be very obvious. So we do highly recommend you to handle with clean, non-powdered gloves, okay, and to avoid contamination with dust. And then make sure that you mix all these staining solutions properly, and then do not be greedy. Stain one blot or one gel per tray. Don't put more than one blot or gel per just to squeeze them together. No, the staining will not be very good. Okay, then also use a high purity water just to avoid the contamination. And if you, let's say, you do not see any signals and low signals, okay, you need to check the pH of this staining step. And then if you using CCD based instrument, you that you image it at a very long exposure time, the stain may fade. Okay, so and because of due to the exposure time, drying, and also and the heating because of the camera itself. So there are different issues contributing to this. And finally, speckled background because of the dust and precipitates. So we will recommend you to filter the buffer and also protect the blot or gel from airborne particles when you do all the staining or even during your Western blotting protocol. So these are the troubleshooting tips. Right, so after you get the data, the total protein data, how should you analyze the data? So I'll be using Azure Spot, which is our free Western blot analysis module, right? So the analytic workflow after the capture is the same, like you use housekeeping protein, right? The signal intensity, signal density for the entire lanes or the large portion of the lane will be used for normalization instead of a single band, right? So here is the interface of Azure Spot, right? We have this 3-plex plus 1 total protein stain over here, the fluorescent westerns. Okay, here we can create a lane. Over here, create a lane. Okay, put them nicely. Then you will see this is channel 4 will be the total protein stain channel. Okay, then now after you put it nicely, the boxes nicely define the lane, you will see this profile. I'll say this signal or the volume here, right? The idea is that you'll be choosing a channel. Here will be channel 4, okay, and also assign lanes that have a defined total protein. This volume of the material in each lane after the background subtraction should be identical if you are having the same protein load across the membrane, right? Their signals should be the same, right? Okay, as you see here, I'll create the lanes. Okay, so I'll just determine the lane, removing the background. Okay, subtract the background. I'll be using rolling ball. There's a rolling ball here, so now you will get these kind of signals. Then proceed to detect the bands of your protein of interest via detect bands, add bands. Okay, now you pinpoint the peaks. Yeah, this is a band I want to quantify, right? Then you see the volume over here. Next, okay, do not detect the bands in your total protein channel. Okay, do not detect the bands on the image. All right. Then when you go to normalization, choose total protein normalization, choose the normalization channel, and also a reference channel. So here's the perks. It works the same as your housekeeping also if you want to analyze it the same way. What you did is you select a lane. For example, I select lane 4 as to be the reference lane. So all the other lane will be normalized relative to this one. Okay, let's say here lane 4, channel 4. Okay, you will see here the normalization factor become 1. This is 1. All these total protein stains volume of the other lanes will be divided by the lane 4 to normalize it. By the end of the day, you will get something we call normalization factor. So here's the equation. Select a lane in the gel or on the plot to be a reference lane. All the other lanes will be normalized relatively to this one, the reference lane. Then for each of these lanes, they will have their own normalization factor, let's say NF. So the calculation is here. Normalization factor equals to the volume of all the materials in this lane divided by all volume of all the material in the reference lane. Simple, right? Right. So now we have the normalization factor. You use this normalization factor to normalize your raw volume of the protein of interest. Here's the calculation. You see here, the normalized volume will be raw volume of the band, here, the raw volume over here, divided by the normalization factor. That's it. Then this is your normalized volume. If you want to do further comparison between like different groups and whatever, then the data you'll be using from this analysis will be normalized volume. Very simple. It will be the same if you use housekeeping protein though, but this case is no longer total protein stains. You need to detect the bands, okay, calculate the normalization factor. It's all available here though. We have different types of normalization in this Azure spot. If you don't want to do normalization, will be click the no normalization. If you want to choose housekeeping, then it will ask you to pick a channel that have the band detected, the protein housekeeping protein bands detected, then and calculate the normalization factor. That's it. Very simple. Okay. Yeah. So this is pretty much how you use analyze it. In fact, you can also do this in ImageJ. Okay. The idea is very simple. Okay, get the lane, okay, get all the volume, okay, all the pixel, all the signal intensity, everything. Then remove the background, okay. You can either choose the background or what kind of different way the background. Then detecting the band, then choose a reference lane, use the other lane divided by this, get the normalization factor. All right, then use the normalized factor to divide your raw volume of the protein interest. That's it. Very simple. If you have any question about this, the calculation, everything, please feel free to email me or ask a question so I can explain further. Or I could have a session with you, trying to type it out and how it works. Okay. Right. So since after you get your total protein image, analyze the data, you get your normalized volume, then you do your comparison between two or more groups. How are you going to put this in your scientific paper, like publish the data? Okay. So this is one of our published data using Azure Red fluorescent total protein stains. On the left, you can see here. Instead of the total or instead of the housekeeping protein that you normally see, like FDH, actin, and so on, this time what you put there is just a total protein stain. And then you just crop an area which shows multiple protein bands. That's it. It's very, very simple. And this is a figure from the supplementary data where you looking at you can see that these are the protein bands, the pattern, okay, in this in the sample as well. So it's very simple. It just replace the total housekeeping protein image that we normally put, okay, instead of just cropping up one single band. All right. So do not worry about the publication. Okay, so we have an application notes that actually tells you how accurate it is, total protein stain when you compare it with the very well-known housekeeping proteins. What we have here is tubulin, beta-actin, and also GAPDH. So this is a fluorescent because fluorescent plot will allow us to omit the variation or variabilities in chemiluminescence method. So it will give us data more reliably. Okay, so we have HeLa cell lysate, 2, 4, 6, 8, 10, 12 microgram, loading them, detecting the total protein, tubulin, beta-actin, and GAPDH, each in their own channels. Okay, and see when we plot the curve or the graph, you see the signals from the total protein is having very good signal linearity, R-square value, than the rest of the housekeeping. Not to say the housekeepings are bad. You can see their R-square is quite good as well, but it's just not as good as the total protein in this case. And how you can also try to verify this is you just different lane have different concentration, right? So if you want to get the raw volume, you should see them the tubulin signal is increasing, okay? if only just using it as a raw volume, like how you use it as a normalization control. But when you normalize it with a total protein stain, you can see that the values remain almost the same. Okay, so just to show you that this is how reliable or the total protein stain in Western blotting. Okay, so let's recap what we have talked about today. First, we talk about housekeeping protein, okay, the drawbacks and alternatives that have been proposing to overcome these kind of drawbacks. Then we finally resolve to the best method, which is the visualizing the total protein after transfer, right? Then since we realized or we found out that that's the best option for now because of cost, okay, cost and time effectiveness, then also not having a lot more consideration to optimize it. And next is how would you choose the total protein stains that fit into your existing or your new protocol. We walk through the staining protocol. Okay, this staining will be different based on the stain, the type of stain itself, and how you analyze the data. The analysis is very simple, just like how you did your housekeeping protein analysis. Okay, then publication-wise is also the same, right? So do not worry about how I want to jump into this trend. I would say the total protein normalization, because everything has, not to say everything, most of the thing has been solved that's provided on the market right now. The benefits and the choices, and also how it fits into your experiment and how you're going to analyze it. There's not really much different except that you are now detecting multiple bands, okay, versus one single band. That's it. And also of your digital imaging requirement. That's quite a few things to consider. Yeah, so thank you so much. I'll open the floor for Q&A session.
Thank you, Kai-Ling. There are questions on the question card. Maybe you want to check.
Yeah, let me put the card out.
Thank you.
No problem. Okay, there are a lot of questions. Oh, some of them have answered. Okay. Right. We have a question for from Azim as well. I'm not sure whether I answered this before. Okay, how would you normalize protein of interest over total protein bands? Correct me if I'm wrong here. It seems to me total protein will stain all the protein of the blot, like how we did with PONCUS. Quite interesting to use total protein as an alternative to housekeeping protein. Yes, the keyword here, okay, like I said, this total protein stain will normally, okay, using fluorescent channel, okay, because fluorescent channel will give you very reliable signals, and then they are very different wavelength, okay. Then here, if you're doing fluorescent Western, it wouldn't be any, I'll say, wouldn't be an issue here, because your protein of interest will be detected in different channel, right? Chemiluminescent also the same thing, yeah. But like I say, in the list of consideration when you choose when you choose your own total stain, will be checking whether this stain will interfere your protein of interest detection or not. Most of the time, it will not happen. But I say, if you really want to check it and use it in your experiment, get a trial kit, test it on your sample, and then see whether it's interfering, or can you see the residual signals on your protein of interest or not. That's pretty quick. Then you see, okay, you can use this or not. I'll say checking or qualification before you really opt for that kind of staining. Okay, yeah. Normalization, I already said it just now. So normalization is the same as how you did on total protein. You just get the raw volume, and then removing the subtract the background, then choose the reference lane, get the normalization factor, then use the normalization factor to normalize your raw volume of a protein of interest. Then finally you get your normalized protein of interest volume. Yeah. That's it. I hope you answer your question. If I miss your or misunderstood your question, please feel free to email me so we can have a in-depth conversation about this. Okay, so well, we have a question from the audience about proteins of nitrocellulose membrane can be degraded after long keeping and room temperature. I personally don't really use nitrocellulose membrane before, but as a common sense that I remember, as much as we have this sample, protein, DNA or anything, we shouldn't keep them in room temperature unless there are specific protocol allows you to keep them or archive them at room temperature. Based on my experience, my personal experience in PVDM membrane, I keep it in PBS, okay, and then keep it at 4 degrees. It can last me for perhaps a year. So after a year, I can take out and image it again. But of course, you have to remember the protein amount might be not, because of the kinetics of it. The signal detection may be not as good as anymore. Yeah. So it also depends on what kind of application they're using. Right. This is quite a personalized question. Okay. Okay. Next question. All right. Chemiluminescence. Sorry, this is popping up. All right. So chemiluminescence Western, the membrane has kept for 2 months in room temperature, then proceed to stain with Ponzu S. The total protein detection is very low. Some protein could not be detected. Oh, so you wonder whether the proteins have been degraded. I do not think that you should keep the proteins or the blot in room temp. I could not really pinpoint what are the factors contributing this. It might be the temperature and it might be the Ponzu S as well. If you look at the Morris paper, the publication that I mentioned just now, okay, the author did mention the limitation of Ponzu S, the limitation of detection in something. So you might need to check it out. I think the easiest way to check this is if you test it with your fresh blot. If let's say you're running a blot right now, you have an unused lane, okay? Then you load the lane with your protein, your positive sample, for example, something you know that is working, okay? Then you cut the part out, straight away send it with Ponzu S total spotting, and then check it whether the stainings are good and the signals are good. So this is a way to try it out, okay, with a fresh blot. So then if it's really that good, then you know that the problem will be lies because of the storage, the storage condition of the blot itself. Yeah, not the staining itself. It might be provide you more insight in this. I hope this will help you. Okay, right. So let me see, do we have any more questions? Okay, I think that we do not have any question anymore.
I think that's from Bun Giat Ma. Do you see the questions?
Hang on, let me scroll up.
Yes, the first question being asked.
Oh, okay. May I know
Thanks.
all Azure systems can with the fluorescence detection capability? Okay, let me go on with the slide. I think it will be easier that way, so I can answer the question. Okay, so yeah, these are the guidebooks and also some other guides. If you're interested into total protein state, how it works and everything, you can check them out. It's all available on our website, or you can ask from Kevin and Ting Ting about this, and then they can send you or direct you to the link. All right. Okay.
These
so
actually in the handout sections, you can actually download them now on the handout sections.
Yeah. All right. Thanks, Nicole. So these you can download them right now in the handouts. Yeah. So to answer Boon, I'm not sure I mentioned the name correctly. So our Azure imaging system comes with 7 models. Okay, we have Azure 200 up to 600. I don't have it here, but you can see that they are ranging in different price. We have the basic gel documentation. The one with fluorescence, we have options of doing only RGB fluorescence or only near infrared fluorescence or near both RGB infrared fluorescence with also the rest of the previous spec like gel door, two types of UV channels and translight, and also the Western CAMI means the CCD, and also true color imaging. Yeah, so I hope this answers your question. Let me put up the question tab again. Yeah, so not all of them having the fluorescence detection is based on the model. So normally if you have model 400, 500, 600 will be fluorescence, having fluorescence module. Yeah. Yeah, okay, so the one, yeah, we have a sapphire biomolecular imager. The one is shown in the, if you join the first two sessions, or in the first image I shown the timeline. Okay, oh, we have it here, sorry. This one, sapphire biomolecular imager. So it is mainly for fluorescence imaging, okay, because it's using 4 laser excitation light sources, and then and also a very good detector for each of the signals. We have PMT, combination of PMT and FPD, which have the best quantum efficiency for fluorescence imaging. Yeah, so this one is higher end, okay, than the Azure imaging system. Azure imaging system is more for Western blotting and a small animal, you know, like very lightweight kind of a detection. But heavy, higher end detection, yes, sapphire biomolecular imager. Okay, so if you have any more question about this fluorescence detection capability, or what the differences between, you know, what are the criteria, then please let us know so we can arrange a session or an online webinar talking specifically about the technologies on this kind of imaging system, why it can do such kind of things. Okay. All right. Let me scroll down.
We have more questions. We have more questions in the question tab.
Okay. Okay. Let me see. We have a question from Putri. Can we proceed for fluorescent Western blot if you use iBind Western system like technology machine? I personally, yeah, I'm not sure about iBind Western blot machine. You might need to check the specs. What kind of excitation light sources they have. Like I say, fluorescence imaging, fluorescent Western blot, you need the excitation light sources and detection as well. So detector, they might be using CCD, but excitation light source, you need to look at whether they have it or not in your system. If your system have the excitation light source, then yes, you can do fluorescence Western blot. Yeah. So that means you can excite it, you can detect it. Yeah. I hope this answers your question.
Yeah, thanks, Kai-Ling. I think Putri, we can actually keep in touch and then talk about this because I think both Kai-Ling and myself, we are not familiar with the iBind Western system. So let us study more and then we can talk further after this session today. Hope it's okay with you. Thank you.
Yeah, we have more question in the question. Okay, firstly, how can we manually determine the total protein intensity? Total protein intensity manually, might know how to say manually means not without the software. Do you mean without the software? Total protein image signals are normally fluorescent signals, so you do need digital software to analyze it. Let's say if you do not want to use like brand specific software, that you, as I said, you can use ImageJ. ImageJ is oh, okay, let me see. There's additional information that without machine. Okay, let me see. Oh, without the fluorescent machine. No, because fluorescent is like this, or total protein stain is like this. You need excitation light sources and the detector. Okay, unlike CAMI. CAMI, because it's a CAMI reaction that generate light, so you just need a CCD camera. But for total protein, you need excitation light sources, and you also need a detector that able to detect it. Yeah. So yes, there's kind of the one of the limitation of using this. So you need to see whether it's compatible with your current equipment or not. Okay.
By the way, to just add in on these questions, so Azure Red could be detected using UV light as well. So if you have a UV system, it's applicable on that. So you can detect it with UV, continue with the experiment, and then you can do your analysis using the software.
Okay.
Sorry for total
Yeah.
it's not available on UV, but Azure Red, yes.
Oh yes. Thanks, Nicole, for telling this. Let me put out the slide here. Yeah. So excitation channel for Azure Red total stain Q and stain free, UV can UV can excite it. So if your imaging system having UV light, so you can excite it and detect it using CCD camera. Yes, you can do that. Okay. Thanks, Nicole, for answering this.
No problem. I think we should wrap up. It's past 4, so.
Oh, okay. All right. Thank you so much, everyone. Let me go to. Sorry. Let me. Yeah. So just like Nicole said, these two guides, the total protein stains and also the ultimate guide to do quantitative Western blot are available in the handouts right now. So you can download them and take a look at it. So my presentation will have just a summary of this or very simplified list. If you want to look for more details, please look at these guides. So it'll be very helpful. Okay. So if you're interested in the total stains, how it works and everything, you can also check out the total stain Q and Azure Red, and then look at their own application note as well, see how it actually works, and then see whether you like it or not. All right. So yeah, we Azure Biosystems, we have imaging solution for every single lab and your application, mostly protein. We have Western blot reagent, AO, also absorbance microplate reader, Azure imaging system with all with basic gel imaging, and also then Western blotting imaging at a higher range, and fluorescence imaging at a higher range. And then we have the highest spec at all, which is a Sapphire biomolecular imager, which has a lot of application right now that is using it at different kind of application, even developing COVID kit and tissue imaging, slide imaging, and also fossil, those kind of things, a lot of them. Yeah. So if you're interested, please feel free to check out. And if you want us to tell you more about this side of the scientific world, which about the techniques and even the scientific techniques, technology, please let us know so we can arrange session, webinar, so we can let you know about it and know what are these machines are really doing with how they capture your data. Okay, thank you so much for joining the workshop. I do hope that you learn something and then take away from this. And yeah, I am Kailing. Please feel free to email us or contact us if you have any inquiries. And we have our Singapore distributor over here, Ting Ting, from Azure Scientific, and from Malaysia we have Epical Scientific, Kevin, and we can follow up on there if you have anything sales or any application inquiries that we can help. Thank you so much. I will see you soon.